longer lesson. Okay, so recording. Okay, it's here. So it started. Yeah, it started. Okay, so let's begin to talk about the eye itself. So first of all, your eye is going to be consistent of the three parts. We have the outermost region. Typically, it is fibrous in consistency. This is going to be the cornea and sclera. Sorry, cornea, as you know, is the thing that is going to be covering this area, right? So it's kind of protruding a bit outwards, while the sclera is this white part that is going to be continuing all the way around the eye, even to the back of it. The vascular component of the eye, this is going to be iris, ciliary body, and choroid. Together, this area is known as the uvea. Okay, so sometimes in the exam, you will see uvea or uveal tract. Okay, so that's also another name that could be used here. Uveal tract is typically talking about the vascular component of the eye. And of course, the most important part of the eye is going to be the retina itself. Retina typically consists of the two regions. We have the anterior part and the posterior part. Posterior part of the retina is, of course, the one which is at the back of the eye. And this is the area which contains your visual neurons, like rods and cones. Maybe from first year, you remember that cones are for the colorful vision, right? And rods are going to be for the black and white kind of vision. The anterior part of the retina is definitely non-visual. So does it contain rods and cones? No. Okay, so the pars iridica, that's iris, and pars ciliaris, that's ciliary body, it does not contain rods and cones, hence it is the non-visual part. The area where anterior and posterior part meet up, this is known as the ora serrata. So, of course, it means absolutely nothing to you. So let's try to see it on the actual anatomy of the eye, right? So we have said that the anterior part, the outermost part of the eye, consists of cornea and sclera, right? So you can see that outermost covering of the eye everywhere, including the anterior region and posterior region of the eye, everywhere the outermost covering is going to be sclera, except for the part where we have our iris, right? So that area, the part of the iris, its most anterior portion, is going to be cornea. You can see that this is the area, so here and here, that's the part where cornea and sclera are continuous with one another. That part is going to have a name, and this is known as the limbus. Okay, so limbus, this is the name of this region. So if I have to specify where is the limbus on this picture, it's here. Because this way, this is the sclera, cornea, right? You can imagine it's kind of bulging forward. So the area where cornea and sclera meet up, this area is known as the limbus. So here's limbus, and this is also going to be limbus. Behind this cornea, behind the cornea, you can see that there is some kind of cavity. This cavity is known as the anterior chamber, and this anterior chamber is filled with the aqueous humor. Aqueous humor is more or less watery, but it does contain lots of gags. Maybe you remember from first year histology, gags are just these negatively charged sugars. And these negatively charged sugars are attracting lots of water. Now, this is the anterior chamber, right? So it is filled with the aqueous humor. Behind the anterior chamber, you can see the colorful part of the eye, which is the iris. The hole is known as the pupil. Behind the pupil, we have the magnifying glass, which is your lens. Lens is attached to the sclera, right? We have established that this outer covering here is the sclera. Lens is attached to the sclera with the help of this area. This area is known as the ciliary body. Okay, so we're gonna call it as the ciliary body. Behind the ciliary body, you can see this serrated area. This serrated area is ora serrata. What does it mean, serrated, right? It means like a saw, yeah? So it has like the sawtooth appearance, something like this. So it's very serrated. So what was the ora serrata doing? As you remember, it was just the area where anterior and posterior parts were meeting up. And you remember that anteriorly to ora serrata, we have the non-visual retina. So what exactly makes up the non-visual retina? The iris itself and the ciliary body. Do you see they're very next to each other? They're within proximity of one another. Just don't forget we're in Hungary, so Latin languages could come up in the exam. So pars iridica, pars ciliaris, it's just in Latin to say iris and ciliary body. So these two areas, they are the non-visual retina. They are considered to be non-visual retina. I think I'm blowing your mind a little bit, but iris is non-visual retina. It has a very similar composition, right? Behind the ora serrata, we have the visual retina, right? So you can see that the innermost covering of the eye, this is the visual retina. That's the area where rods and cones are going to be sitting. But what is between the visual retina and sclera? Don't forget that the most outer covering everywhere, doesn't matter anterior or posterior part of the eye, is always sclera. So what is between sclera and retina? We have choroid. In this picture, it's very nicely showing you that choroid is the vascular part. Do you see there are lots and lots and lots of blood vessels here? So choroid is the vascular part. Now, behind the lens or behind the ora serrata, we have this body which is called vitreous body. And vitreous body is sort of to the inner part of the retina, right? So it's under the retina. Vitreous body is jelly. It's compared to the aqueous humor, right? It's more gelatin-like. Why? Because that's the body, that's the humor that contains the most of hyaluronan. And I'm not sure if you remember, but hyaluronan is the biggest gag. It is a gag, it is a glycosaminoglycan, but it is the biggest gag there is. If it is the biggest gag there is, it attracts lots of water. So much water and so much hyaluronan, which I repeat is a gag is a sugar, creates this jelly-like appearance. Yeah, so the reason why vitreous humor is so jelly-like, so gelatinous, is because of the hyaluronan. What is the purpose of the vitreous humor? To make sure that the eye does not collapse, right? So it fills up this entire core of the eye to make sure that the eye definitely stays quite open, right? So it doesn't collapse. Okay, we said there is anterior chamber, right? Anterior chamber is in front of the iris, but there is also posterior chamber, and posterior chamber is behind the iris, but in front of the ciliary body, right? Do you see there's a little bit of space right here, which is between the iris and ciliary body. That's the area where posterior chamber will be. What is the importance of posterior chamber? This is the area where aqueous humor will be produced. So we make aqueous humor, we make this watery substance in the posterior chamber. Who do you think will be making this aqueous humor? The ciliary body. Okay, so the ciliary body. Ciliary body is going to produce the aqueous humor, put it into the posterior chamber, and then push it through the pupil to the anterior chamber. So the aqueous humor will be flowing like this, from the posterior chamber to the pupil, to the anterior chamber. And then there's a bit more details. We'll talk about it. Is it clear? At the back of the eye, here you have an optic disc. Optic disc is considered to be a blind spot because this is the area where rods and cones are absent. So in this part of the retina, you will not find rods and cones. So that's why it's a blind spot of the eye. Optic disc is the beginning of the optic nerve. So that's the area where some kind of axons are going to be coming here. Now, what the hell? Let's talk about which kind of axons. So we said in retina, there are rods and cones. They are photoreceptors. Their axons are synapsing with a cell in the retina. The cell is called ganglionic cell. So there is a ganglionic cell sitting in the retina. It receives impulse from the rods and cones. The axons of the ganglionic cells go all the way here to the optic disc and make up optic nerve. So basically, the entire optic nerve consists of axons of ganglionic cells. But where the ganglionic cells are sitting? In the retina itself. So you can imagine that these cells, they have a very long axons, right? So super long axons that are just going out. And you remember that the axons, the optic nerve, it will go through the thalamus to the occipital lobe, right? To make sure that you can see things. There's additional area here, which is known as the fovea centralis. As you can see, it's a bit this way, right? So it's a bit next to the optic disc, this fovea centralis. Another name for it, it is also known as the central depression or central pit. Okay, so another name for it, it is going to be central pit. The purpose of the fovea centralis is that it only possesses cones. So you will not find rods in this central pit. You only have cones. And the purpose of fovea centralis is the central vision, no? Tunnel vision, okay? Central or tunnel vision. For sure, you have experienced this tunnel vision once in your life. For me, that's typically when I'm watching, like, I don't know, something I like on TV, like Game of Thrones, right? Or something like this. You cannot see anything. In your periphery, you cannot see. You can only see what is in front of you. Another example of tunnel vision is when you're driving. If you know how to drive, you have experienced this. So when you're driving, you can only see what is in front of you. You cannot see the periphery. Or when you're reading. Basically, when you're super focused on one thing. Okay, so when your eyes are super focused on one particular thing, that's when your fovea centralis is on fire. So it is working. You're definitely seeing, like, you're experiencing the tunnel vision. The tunnel vision, I repeat, is when your periphery, right, it's non-visible. It becomes almost black, that it's not there. You cannot see it. You can only see what is ahead of you. You can actually experiment it, right? So just start looking at one thing and really focus on it. And with time, you will realize that you cannot see anything on the side. You cannot see there. That's tunnel vision. So that's the general anatomy of the eye. So now let's take a look a little bit more details into each part. So we start with the cornea. Cornea is, as you remember, it's this, right? So it's this area right here. It's the part which is covering your iris. Behind the cornea, we have the anterior chamber filled with aqueous humor. Cornea is avascular structure, doesn't have any blood vessels in it, and it consists of 5 layers. The outermost layer, the one which is facing outside, is the stratified squamous non-keratinized epithelium. And this is very interestingly because skin is facing outside, but it is keratinized. Your cornea is also facing outside, right? It's the air itself, the outermost layer that is facing outside, yet it is non-keratinized. Why is it non-keratinized? Because it contains lots and lots and lots of nerve endings. So within the epithelium, within this area, there are lots of nerve endings. In physio, if we weren't during the pandemic, in the middle of pandemic, in physio you had a lab, you would have had a lab uh, where you take like a Q-tip, right? And you try to swipe the cornea. You just try to touch it. And what happens as a result? Your eyelids, they close. It's a palpebral reflex, right? So palpebral are your eyelids. So palpebral reflex, it means that if you swipe the cornea with something cotton, right? Like your Q-tip, for example, your eyes will shut down. Why? Because of these nerve endings. So these nerve endings are starting in the outer layer of the cornea, but they're going all the way to your brain. Okay, so to kind of create that kind of reflex. Under this layer, we have anterior limiting membrane. Another name is Bowman's. It is a non-cellular, so as a result, it cannot be regenerated. So if, like, I don't know, you damage this layer, it will leave a scar. Okay, so it will leave a scar. So you'll need to replace the cornea. Stroma is the thickest layer. It contains keratocytes. Keratocytes are basically fibroblasts, basically. So fibroblast in the cornea is known as the keratocyte. From histo 1, you remember that fibroblasts are responsible for secreting the entire extracellular matrix. So you're going to see that here specifically in the stroma of the cornea, these keratocytes, they're secreting collagen type 1. What is another place for collagen type 1? Skin and bone. So now you're going to add the third place, which is cornea. So type 1 is the thickest, strongest, baddest collagen there is. So it's needed here, right? Because cornea is facing outside. We want it there to make sure that it is protected. Posterior limiting membrane is known as the deciment. It is non-cellular as well as the anterior one, but it can regenerate. Why? Because of endothelium, the one layer under it. These endothelial cells here are super special. For some reason, nobody knows how exactly, they are partially stem cells, so they can give rise to this area as well. So they can secrete the collagen fibers in this area as well. So the main difference between Bowman's and deciment, one cannot regenerate, one can. Deciment can regenerate because of the underlying endothelium. This endothelium is super special somehow. Okay, so nobody knows how exactly is it possible that it regenerates the deciment, but they know that it's definitely because of this endothelium. So this is just additional information. I want you to read it at home. Okay, so it's just additional information about each of these layers, but the most important things we have covered. I want to get your attention to here. So stroma, the third layer of the cornea, is the thickest part of the cornea, and it consists mainly of water. Okay, so it's very watery. The collagen type 1, which is located in the stroma, is arranged parallel to each other. Very, very parallel to each other. Plus the water content, plus the spaces between the collagen type 1 fibers, it creates this transparency that is needed for cornea, right? Cornea is very transparent. I can see everything through it. It's very transparent. Yet collagen type 1 is the thickest collagen there is. So if I simply put a bunch of collagen type 1s together, I'll make skin and bone, which is not transparent. So how is it that cornea is transparent even though it has the same collagen type 1 in it? I repeat, the arrangement is different. So these collagen type 1s in my cornea are parallel to each other. They have spaces between each other and super high water content. Skin and bone is not this watery. Okay, so the same, right? So epithelium, that's your cornea. If you haven't understood what we're talking about this area right here. So that's your cornea, the most anterior part of your eye. It has 5 layers. Epithelium, don't forget stratified squamous non-keratinized epithelium. It has the anterior limiting membrane beneath it, which is the Bowman's. It has stroma. The cells in it are called keratocytes. They are fibroblasts. They're responsible for secreting this entire stroma. We have posterior limiting membrane, which is the Descendens membrane. And beneath it, we have the endothelium. What is going to be here?
The anterior chamber.
Very good. That's where the anterior chamber will be. So beneath the cornea, we have the anterior chamber. And what is within this anterior chamber?
Aqueous humor.
Aqueous humor. Okay, so this is the anterior chamber. This is the iris, as you can see. So this space right here behind the iris, this is the posterior chamber. So this area right here, that's the posterior chamber. The way we're going to do our class, we're going to look at each structure first, and then at the end of the class, we're going to look at the whole slide. Okay, so we aren't doing theory first and then the whole slide. We're going to look at every structure little bit by little bit because there's so many things on the eye, so it will be too much. So you can see that this is your whole eye. So this area right here is the cornea itself. What is this huge bulb?
Lens.
The lens.
This is going to be lens, right? And you can see that this is the cornea itself. So let's quickly do the layers. This is
Stratified squamous non-keratinizing.
non-keratinized epithelium. As you can see, it's not very thick. It's relatively thin. That's where the external environment is, right? So that's where the air is. What is this?
Anterior limiting membrane.
Anterior limiting membrane, Bowman's membrane. From here, approximately until this area, that's where the stroma is. What are these?
Collagen type 1.
Collagen type 1. These elongated nuclei belong to keratocytes, which are basically your fibroblasts. Notice there are spaces here. There are lots of spaces, and these spaces aren't artifacts. They aren't. They are needed for the normal transparency of the cornea. Okay. So this is the posterior limiting membrane, which is the Descendens membrane, and this is the endothelial cells, right? So the endothelium. What do we have here? This is the anterior chamber. As we have said, anterior chamber is filled with the aqueous humor. The entire cornea is avascular. So could you tell me from where does it get its nutrition?
From the aqueous humor.
Very good. Half of it, exactly half of it, will get its nutrition from aqueous humor. Exactly half of it will get its nutrition from aqueous humor. So thanks to the endothelial cells, they'll be able to absorb a little bit of the aqueous humor and give the nutrition to this area. But what about the more superficial part? What is on the surface of my eye? Always should be there, at least. Tears. During the exam period, there are more of it, but in relatively normal lifestyle, less, but tears. And these tears are very important because they will definitely supply the superficial part of the cornea with nutrition. Yeah, so two sources for nutrition of cornea: the tears and the aqueous humor. Okay, conjunctiva. Okay, so this is additional tissue which can be present on the surface of your eye. Okay, so we have said that this is the cornea. This is the sclera. The area where cornea and sclera meet up, this is called limbus, as you remember, right? So that's the area where cornea will continue as the sclera. But on the surface of the sclera, right on the surface of the sclera, we have conjunctiva. And this conjunctiva is known as the bulbar conjunctiva. This is the eyelid itself, right? This is the inner part of the eyelid. And you can see that there is another conjunctiva there, which is called palpebral. Conjunctiva is basically epithelium. That's what it is. It's nothing more. It's just a bunch of epithelial cells. But there is one more additional thing that you need to remember, is that there is a border where bulbar conjunctiva meets up with the palpebral. And I chose a wrong word. It doesn't meet up. It reflects on this area. This area is known as the fornix. Okay, so fornix is the part where bulbar reflects and becomes palpebral. Because think about it, if we draw a line, if we draw the conjunctiva, right? So where is it? So it's this part. Do you see? This is your sclera. And you can see that right on the surface of the sclera, there is this conjunctiva. Which one is this?
Palpebral.
Palpebral is on the eyelid. So this one is
Bulbar.
bulbar. So this little tissue that you can see here, this is bulbar conjunctiva. And do you see it suddenly abrupt, like it stops. It abruptly just stops. That's it. Why? Because we're missing a very important part here, which is your eyelid with the eyelashes. Let's draw eyelashes here like that, so you make understanding. This is the eyelid itself, this tissue. This is the eyelid itself. The inner part of the eyelid, the inner part, is also covered by conjunctiva. Which one?
Palpebral.
Palpebral. So how do you call this area right here?
Fornix.
Fornix. In our slide, we have something like that. We don't have eyelid in the tissue. We don't have it. So the area where you see that it just suddenly stops, do you see? Suddenly stops, that's the fornix. Because from this point, you should have reflected onto the surface of the eyelid, right? Where the palpebral conjunctiva would be. More eyelashes here. So do you get it? Limbus, we talked. Okay, so take a look where we are. So let's take a look. What was this? I know it's very little, but what is it?
Cornea.
Cornea. How do we call the cavity behind it?
Anterior chamber.
Anterior chamber. So take a look where the little square is, right? So what are we looking at? What is this? The most superficial part. Take a look where the square is. How do we call? What is this?
Bulbar conjunctiva.
Very good. So this entire thing, this epithelium that you see here, as you can see, it is stratified squamous epithelium, right? So maybe even cuboidal a little bit in this region. So stratified squamous, stratified cuboidal epithelium, this is bulbar conjunctiva. What is this? What is bulbar conjunctiva on?
Sclera.
Sclera. So what you see, this pinkish tissue right here, this is sclera. What do you think this is?
Ciliary muscle.
Probably ciliary something, right? So we could say so, but it's not ciliary, it's actually already retina, because we can see many layers. Do you see where we are? So iris is this line right here. This is iris. Ciliary body is this little tiny dot. Do you see where the square? It's a bit more at the top of the ciliary body. So what you see here, this is already retina, already visual retina. And how do we call this between sclera and visual retina? We called it choroid. And what was so special about choroid? That's where the blood vessels are. So how do you call this abruption right here? Of course, it has to be
Fornix.
fornix. So that's the area where you would have reflected onto the surface of the eyelid. And that's where the palpebral conjunctiva would have been. But we don't have an eyelid. Do you see? We just have an eye. We don't have an actual eyelid on the slide.
Miss?
Yeah.
In the previous, in this over here, can we see the ora serrata?
Very good question. Not really. Okay, so not really. Ora serrata is more visible on the actual eye, but on our slide, the vitreous humor, which was supposed to be here, it was drained. So as a result, ora serrata also kind of collapsed. So they just removed it. Ora serrata is more visible on the actual eye, not the sliced one. And this one is sliced. So we cannot really see it. It has collapsed.
Thank you.
Okay, so the same idea, right? So this is conjunctiva, this is sclera. So you can see that this is the bulbar conjunctiva. It suddenly stops. The point where it suddenly stops, that's the point where reflection should have been, right? So the area where it should reflect, this is known as the fornix. Okay, let's talk about ciliary body. So again, maybe a little bit of anatomy, because anatomy is very important for the eye, right? So this area right here, this is your cornea. What is here?
Anterior chamber.
Anterior chamber. You can see that this is the part where cornea becomes what?
Sclera.
This is sclera, yes. On the surface of the sclera, what should be here?
Conjunctiva.
Which one?
Bulbar.
Bulbar. Specifically, bulbar conjunctiva should be here on the surface of the sclera. Behind the anterior chamber, that's the area where iris will be. The hole in the iris is known as the pupil. Behind of pupil, you see lens, the magnifying glass, right? How do we attach lens to the sclera? With the help of the ciliary body. Ciliary body is kind of a very interesting structure because it consists of two things. It consists of the ciliary muscle, which is going to be somewhere here. That's ciliary muscle. And on the surface of the ciliary muscle, you will see ciliary processes. Ciliary processes are just epithelial cells. And these epithelial cells have one job: to secrete aqueous humor. To secrete it where? Into the cavity right here behind the iris, which is the posterior chamber. So I repeat, ciliary body consists of two things: ciliary muscle and ciliary processes. Ciliary processes are these epithelial protrusions. These ciliary epithelial protrusions are responsible for secretion of aqueous humor into the posterior chamber right here. What is the ciliary muscle responsible for? It is responsible for controlling the shape of the lens. We have established that lenses are attached to the ciliary body itself, right? So if this muscle is contracting or relaxing, because lenses are attached to it, the lens will also change its shape. How do we call the connection of the lens to the ciliary body? We call it suspensory ligaments, zonular fibers, or fibers of Zinn. Three names for the same thing. Okay, so the ligaments which are attaching lens to the ciliary body itself, they're known as the suspensory ligaments, zonular fibers, or fibers of Zinn. So, ciliary body, I think it's very confusing for lots of people, especially if we always see picture like this. So what is this?
Iris.
Iris. What is this chamber? This is the posterior chamber, and this is the ciliary body. This is the lens, and what is this?
Suspensory ligament.
Suspensory ligament. Very important to remember that ciliary body is actually a ring shape structure. On this picture, it's very difficult to imagine, but take a look here. That's ciliary body, right? So it kind of imagine iris is also like a ring, right? Iris is also a ring. But on this section, we see iris as two parts, just this part and this part. The same is for the ciliary body. On this picture, we see it just like the upper region and the bottom region. But in reality, ciliary body is a ring as well. It's a ring around the lens, literally. It's kind of enveloping, how do you say it? Like it's fully coats the lens on the outer part of it. Does it make sense? So it's not like a upper and a lower kind of structure. It's not just two of them at the top and at the bottom, what we can see on this picture, right? There's a top ciliary body and there's a bottom ciliary body. No, it's a ring. It's a ring around the lens itself. Okay, let's talk about this part. For sure, you're gonna see a question, for sure. So a little bit of the anatomy again. So this is cornea. This is the anterior chamber. This is the iris. This is the lens. This is the ciliary body. These are the suspensory ligaments which are attaching to the lens. We said that ciliary body consists of the processes, right? So the ones which you can see here, the epithelial processes at the top, and the muscle itself. So let's imagine something like this. If the ciliary muscle contracts, right? So if this muscle contracts, what's going to be happening to your, okay, how to rephrase it? If it contracts, does it come closer to the lens or away from the lens?
Away.
In reality, I could see, like, that's the confusing part. I could see, like, in my 5 years of tutoring experiences, every year is the same thing. So it doesn't matter how even I try to explain it. Whenever I ask this question, if the ciliary muscle, if it contracts, does it come closer or away from the lens? Every single person will say away, including me. The problem is, it's that it contracts, okay? So it comes a bit closer. When this muscle contracts, it comes a bit closer to the lens from two sides. Imagine it's a ring, and this ring is contracting. It's contracting. So does it come closer to the lens or away? Definitely closer, because it is a ring-like structure. This ring is contracting, right? So it's coming closer to the lens. If this ciliary muscle comes closer to the lens, the suspensory ligaments, are they stretched or relaxed?
Relaxed.
They're relaxed. So they aren't pulling on the lens. If suspensory ligaments aren't pulling on the lens, the thickness of the lens increases or decreases? Increases, right? So basically, can you imagine what's happening? Ciliary muscle is contracting, right? It contracts. The ligaments aren't stretched. You're basically increasing the thickness of the lens. You're basically making a magnifying glass. You're increasing the thickness. You're making a magnifying glass. What is this magnifying glass needed for? For close vision. Think like a microscope, right? I'm increasing the thickness so I can see more up close. So if ciliary muscle contracts, that means that you're focusing on the close point. The opposite is for the distant vision. If your ciliary muscle relaxes, it gets away from the lens. What happens to the suspensory ligaments? They start to stretch As they stretch, they pull the lens become much thinner, so you can see more distant. And now let's do experiment. I want you to take your finger and put it very close to your eyes, very, very close to your eyes, and try to focus on it. Like try to focus and then look away. Try to focus on it and then look away. Do you feel the tension as you're trying to focus on the eye which is up front? Do you feel the tension in your eyes? That tension is the contraction of the ciliary muscles. They're super, super busy contracting to focus on what you're looking at, and they're relaxing as you're looking away, right? You can actually feel the relaxation as you're looking away. So remember that to see something close, the ciliary muscles have to contract. As they contract, they have to make the lens thicker so you can actually see what's in front of you. Make sense? Okay. So let's talk about ciliary processes. So you can see that this is the lens. This is the suspensory ligaments, zonula fibers of Zinn. Somewhere here would ciliary muscle would be, right? And what we can see here, this is a ciliary process. We have said that ciliary process consists of epithelium, right? Basically, it is an epithelium-like structure. There are two layers of epithelium, pigmented epithelium and non-pigmented epithelium. The bottom layer is pigmented epithelial cells. The top are, of course, non-pigmented. To make, what was the job of this ciliary processes? To secrete aqueous humor into the posterior chamber, into somewhere here. So how exactly can they do so? What do we need to secrete the aqueous humor? We need blood. We're gonna take plasma of the blood and convert it into the aqueous humor. So ciliary processes are very vascular. They're filled with blood vessels, right? So these cells, they're going to take up the plasma of the blood vessel and convert it into the aqueous humor. But what I want from you to understand is the following. So we have said that this is the ciliary body. Here are the ciliary processes. In the ciliary processes, the blood vessels would be, right? In the ciliary processes. What is the purpose of the ciliary processes? Blood vessels to secrete aqueous humor into the posterior chamber. The big question that they're going to ask you here is, whose blood vessels are in the ciliary processes? And what's important to remember is that we don't have like a ciliary artery that is going to branch out and give me branches in the ciliary process. We don't have that. The blood vessels that are located in the ciliary processes are branches of the artery of the iris. So we have an arteries in the iris, and they send branches into the ciliary processes. Is it clear? Yes, no? So somewhere here we'll have the iris itself, right? It has the blood vessels, the arteries of the iris. They go and give branches to the arteries of the ciliary processes. Okay. So speaking of ciliary muscle, because it is a smooth muscle, right? So first of all, it's not a skeletal muscle. I can't control it. So it is a smooth muscle. What's up with its innervation? So innervation is going to be parasympathetic, right? And sympathetic. Sympathetic is very easy. It's via the long ciliary nerves. What is the purpose of the sympathetic innervation of the ciliary body? They did a test, right? So if you're startled, like you're suddenly, like, I don't know, surprised by something, you're going to see that your eyes will automatically adjust for distance vision. The job of the sympathetic innervation is going to be to relax your ciliary muscle, distance vision. job of the sympathetic innervation via this to relax the ciliary muscle so you can see more far away. And think about it, sympathetic is fight or flight, right? And to fight, I need to see what's far away. I need to make sure that nobody is hunting me right now. So of course, it's going to be the far away vision. Parasympathetic is more for a closed kind of vision. As you remember, here we're contracting muscle for the closed up vision. So parasympathetic will be originating is this Edinger-Westphal nucleus. I have a homework for you guys for the next class, research about this one. I'm going to ask you all the functions of this nucleus. They love talking about this nucleus, both in neuroanatomy and in neurohisto and in neurobio, in everything. Why? Because it's the most researched nucleus there is. It's the only nucleus that we actually know pretty much everything about it. The rest of the nuclei we do not know most of the things, okay? This is the most researched nucleus, so they'll for sure ask you about it. Okay, aqueous humor. So, very important question, what is the pathway? So where exactly is it produced and how does it flow? So let's start. Okay, so who makes aqueous humor?
Ciliary body.
Ciliary processes of the ciliary body, right? They're going to be somewhere here. So ciliary processes are going to be secreting the aqueous humor and putting it into this chamber right here. Which chamber is this?
Posterior chamber.
Posterior chamber, between the iris, right, and between the lens itself and ciliary body as well. The aqueous humor starts to flow from the posterior chamber into the pupil, right? So you can see that the next one is the pupil. We start posterior chamber, then pupil. From the pupil, the aqueous humor starts to flow into the anterior chamber. So you can see that this is the third place. From the anterior chamber, it flows into this angle right here. This angle is between your iris and cornea. So as a result, this angle is called iridicocorneal angle or iridicocorneal corner. So that's the area where the aqueous humor starts to flow. I'm going to magnify this way so we can actually see, right? So where we go. So this is the iridicocorneal angle right here. We said that aqueous humor goes from the anterior chamber all the way to this corner. From here, the aqueous humor starts to flow into the spaces of Fontana. Okay, you're gonna see that there will be some kind of gaps in this area. We're gonna call them spaces of Fontana. From spaces of Fontana, it goes into the endothelial anastomosis. I apologize, endothelial anastomosis, which is typically located somewhere in the sclera. Okay, so it's very wrongly drawn here. Okay, so here you're going to see that there is some kind of trabecular meshwork. It's the same name for endothelial anastomosis, so it's somewhere in the scleral portion. From endothelial anastomosis, it goes to the canal of Schlemm. In this picture, they always draw canal of Schlemm in the cornea, right? That it belongs to cornea, this canal of Schlemm. In ours, it's in the sclera. In our slide, canal of Schlemm is in the sclera. I go back because, no, I go, where do I go? No, I don't go here. I go here. So these are canals of Schlemm, right? You're going to see that they always show canals of Schlemm as part of the cornea, but in our slide, it's more in the sclera itself. From the canal of Schlemm, you're going to see that it's going to flow through some kind of ocular veins until it reaches the central vein. Where are the ocular veins? They are somewhere in between sclera and choroid. So from canal of Schlemm, the aqueous humor starts to flow in this way, in the ocular veins between sclera and choroid, until it reaches the central vein of retina. Central vein of retina is here. It's in the middle of the optic nerve and optic disc. Okay, you're going to see that that's where the central vein will be. So do you see, eventually, all of this aqueous humor ends up in the vein. It ends up as part of the vein. So it's very important for the aqueous humor to not stagnate. It shouldn't accumulate in the anterior chamber. It should always flow towards the central vein. As previously, I think I said, it's very important. They ask, okay? So they ask this whole pathway. So exactly as it was written here. Okay, so let's take a look where we are, right? So little box is here. So again, what is this?
Cornea.
Cornea, anterior chamber lens. What is this line?
Iris.
Iris. And what is the box pointing at? What is this?
Ciliary body.
Very good. I will show you where ciliary body is. It's this with this. So together, they're ciliary body, together. What does ciliary body consist of? It consists of this. What is that?
Muscles.
Ciliary muscle. And it consists of this, which are ciliary processes. Notice how vascular the ciliary processes are, right? You can see lots of blood vessels in it. What is this?
Suspensory ligament.
Iris.
Iris, very good. This is iris. It's a little part of iris. So these blood vessels are originating from the iris, right? We can actually see how blood vessels are coming from the iris into the ciliary processes. What is this?
Clara.
A little bit this way, it's sclera. A little bit this way, it's cornea. Do you see? It's like a junction point. Take a look where we are. It's a junction point. So how do we call the junction point?
Limbus.
Limbus, yeah. So the more correct way would be that this is limbus. So what you can see here, this is the limbus. It's the like a transition zone where cornea transitions into the sclera. So the top, this is the sclera region. The bottom, this is the cornea. This, exactly this, is the limbus. Let's magnify into the ciliary processes, right? We can see the top layer is non-pigmented, the inner layer is pigmented. So these are the two layers of the ciliary processes with a bunch of blood vessels present. I repeat, these blood vessels belong to iris. Yes. What is this? This is lens. It's a little piece of lens visible here. This is the capsule of the lens. And what are these?
Gag.
Sorry?
GAG.
Again?
Gag.
Not gags. These are, yes, these are suspensory ligaments. They're just broken down. Okay, so these are what you see here floating. These are the suspensory ligaments. They should be attaching to the lens, but during preparation, they were kind of damaged. Okay, so you can see that these are the suspensory ligaments. Okay, blood ocular barrier. So basically we have blood aqueous barrier and blood retinal barrier. Blood retinal barrier are going to be non-fenestrated capillaries of the retinal circulation together with the tight junction of the retinal epithelial cells. So blood retinal barrier is going to be somewhere here, okay, or here. We're going to see that on the top of the retina, we have the choroid plexus. Choroid plexus is very vascular. Right under the choroid plexus, there will be a little bit of the retinal blood vessels. These blood vessels are non-fenestrated. Right under the, what do I mean by non-fenestrated? I mean continuous. It's just your normal continuous capillary, what we have learned in the first year. Right under the blood retinal barrier, you're going to see some retinal pigmented epithelial cells, they have a bunch of tight junctions. Okay, it's just two layers here. The continuous capillary right under retinal epithelial tight junction areas, right? Blood aqueous barrier is between ciliary epithelium and capillaries of the iris. What do I mean by capillary of the iris? I mean these. These are considered to be capillaries of the iris. Why? Because they are branches of the arteries of the iris. So the blood aqueous barrier is right here. Blood aqueous barrier is right here. Okay, we aren't allowing the blood to go this way, and we aren't allowing the aqueous humor to go back way as well. Okay, so we aren't, that's where the blood aqueous barrier would be. Inflammation can break down the barriers, right? Both the retinal and the aqueous. So because cells get damaged, so it's not good. Speaking of iris, iris has two layers. It has the top stroma layer and it has the bottom epithelium. So the more anterior layer of the iris is stroma. The more posterior is epithelium. That should like kind of make you question things, right? Because typically the most anterior is always epithelium. The most top is always epithelium. Here it's the opposite. Okay, you see that epithelium is actually the most under tissue, while the stroma is the most anterior. The epithelium is pigmented, and this pigmented epithelium is responsible for the color of the eye, right? What is the purpose? Absorb the light, to prevent the refraction of the light, to make sure that it doesn't scatter, that it doesn't break, and etc. Within the stroma of the iris, we have two muscles. We have the sphincter muscle and dilator muscle. What do? Well, sphincter muscle will be constricting the pupil to make the pupil smaller. The dilator muscle will make the pupil larger. So what's the innervation? Parasympathetic from Edinger-Westphal, sympathetic from superior cervical ganglion. Very important. Okay, so you can see that it's a bit the opposite, right? Here, constriction of the smooth muscle, of the sphincter muscle, is parasympathetic. And relaxation, right, dilator, you're gonna see that it's actually sympathetic. Iris is divided into two zones. We have the pupillary zone and we have the ciliary zone. If you have light eyes, you can actually see this area, which is known as the collarette. So in our first slide, the picture I put you there is a light eye. You can actually see it there. You can see the junction point. What is collarette? Collarette is the remnant of the coating of embryonic pupil. So basically, this pupillary zone was added later, like much later. In embryo, the pupil was until here. It was much bigger. Okay, so in embryo, the pupils are much bigger. So the pupillary zone of the iris was added later. So in the light eyes, in the brown, maybe if you like shine the light directly into the eye, in the brown eyes, you might be able to see the collarette, but in the light eyes, you can see it like immediately when you look into the person's eyes. It's just embryonic structure, nothing interesting, yeah? So take a look where we are. So this whole thing is iris. Tell me, what is this?
Cornea.
Cornea. What is this?
Anterior chamber.
Anterior chamber. This is the iris itself. This?
Lens.
Lens. And this is the capsule of the lens. It should be attached to the surface, to the anterior surface of the lens, right? So you should be able to see it there, but you can see it's a bit detached. So what is this space here?
Posterior chamber.
Posterior chamber. What does the iris itself consist of? Well, you can see anteriorly we have stroma. It's highly vascularized. Posteriorly we have pigmented epithelium. right? So probably the color of, I'm not sure if it is a rat or if it is a human eye, but the color was brown, right? So you can actually see it a little bit. So how about the muscles? So until here, this is the sphincter muscle, and here, this is the dilator muscle. This muscle will be contracting this way. This will be contracting that way. So as a result, the pupil, right? So this gap right here, its diameter will be changing. Edinger-Westphal, Edinger-Westphal, and superior cervical ganglion. That's the innervation for these two muscles. So again, how do we call this?
Cornea.
Cornea, anterior chamber, iris, posterior chamber. Stroma, pigmented epithelium. What are these?
Ciliary process.
Very good, with two epithelial cells as well, right? So the top are non-pigmented, the bottom are pigmented with lots of blood vessels. This ciliary muscle, right? So this is the cornea. This is already the sclera right here. So this region, this weird like transitional region, that's where the limbus would be, right? Okay, ciliary muscle we talked about. It's just additional information to make sure that you understand how it works, right? So that it is a circular structure. Just read it at home. Okay, this slide is to show you the pathway of the aqueous humor. So these ciliary processes start making the aqueous humor and put it into the posterior chamber. So this is the posterior chamber right here. From here, the aqueous humor will be flowing to the pupil. You cannot see it on our slide here, right? But it's gonna go to the pupil and then end up in the anterior chamber right here. We have said that this is the iris itself. This is the cornea. So from the anterior chamber, the aqueous humor flows into this angle. How do we call this angle?
Iridocorneal angle.
Very good. Yes, we call it iridicocorneal angle, right? It's the angle or corner between iris and cornea itself. So it flows into this angle. From iridicocorneal angle, it's going to flow into these spaces. Do you see? This is a space. This is also a space. And this is also a space. So these are spaces of Fontana. So from iridicocorneal angle, it flows into the spaces of Fontana. From spaces of Fontana, it flows to?
Canal of...
Before canal of Schlemm, there is endothelial anastomosis. Okay, we cannot really see it. Okay, we cannot see. So somewhere here, they always point that maybe these are endothelial anastomosis. But then it goes to the canal of Schlemm. Okay, so these two right here, they are canals of Schlemm. From canals of Schlemm, it flows into the ocular veins. So these right here, these branches, they are ocular veins. Ocular veins are going all the way this way, right? So there are going to be a bunch of them here, ocular veins, until eventually from here, it goes to this region right here, where optic disc will be. That's where the central retinal vein would be, right? So the aqueous humor from these ocular veins for the entire eye are going to be going to the central retinal vein somewhere here. Is it clear?
Yes.
There is something called glaucoma, and glaucoma is when you have buildup of the aqueous humor in the anterior chamber. As a result, it pushes all the tissues to the back. The problem is that at the back we have the optic disc, the area where optic nerve would be. The vessels here become compressed. That causes necrosis, death. Your optic nerve starts to die. It leads to blindness. Uncontrolled glaucoma leads to blindness. What is one of the reasons for glaucoma? Well, there must be a problem here, right? So canal of Schlemm, probably. It can't drain aqueous humor into the oculus vein somewhere here. So there is like a occlusion of canal of Schlemm. Why? Sugar. When you have high sugar, it starts to build up in weird places. One of them is here. So glaucoma is actually very much related to diabetes. If you have diabetes, you most probably will have glaucoma. So you need to control it. How exactly? By blocking M3 receptors, muscarinic receptors. These muscarinic receptors are present on the ciliary processes, on the epithelial cells of ciliary processes. If you block them, the ciliary processes won't make as much of aqueous humor. They won't. The little aqueous humor production will release the glaucoma, and yeah, like it will suppress it, right? It will make it better. Fun fact, marijuana can be used to treat glaucoma. It's one of the most used, how do you call it, like excuses in US and Canada, right? So you can buy the prescription for marijuana and say that you have glaucoma. So it's one of the treatments, because marijuana will also be blocking the M3 receptors here specifically. Hydrostatic pressure, right? So that's the most important thing. So aqueous humor is not very proteiny. It doesn't have much proteins in it. So when it builds up, it causes a rise in hydrostatic pressure. There's too much of it. Lens. Lens are basically a bunch of proteins together, just stuck up together. The protein is called crystalline. It has a very particular shape, a very particular arrangement. And if something happens to crystalline, if it has some kind of conformational change, it won't be the same crystalline. So that causes cataract. Cataract is when the lens are cloudy. Yeah, they aren't clear. You can see that they're cloudy. There's like a white discoloration in the lens. Do you think it's easy to see with the white discoloration in the lens? Of course not. Lenses have to be transparent. So cataract is blindness, right? So you can see the shapes, but you cannot see very clearly. So cataract happens, I repeat, because crystalline, the protein that we have in it, it changes. It has some kind of conformational change. What could be the reason for conformational change of crystalline? Well, so many different reasons. It could be genetical, it could be diabetes, certain drugs can cause cataract, okay? So it's all like, the reasons are unlimited. So there are lots of reasons for cataract development. Now, there's the question that they always ask. If you look at the lens, it has a capsule. And the capsule is going to be present only on the anterior surface of the lens. Okay, so you're only going to see capsule on anterior surface of the lens. Anterior surface of the lens is facing posterior chamber. So that's where the confusion could be. Okay, so I repeat, the capsule is only present on anterior surface of the lens, which is facing posterior chamber. So take a look, right? So this is the lens. What is this?
Iris.
Iris, the pigmented epithelium, stroma. This is the posterior chamber behind it. What is this?
Capsule.
Capsule.
Excellent.
Follow the capsule. You see, it stops here. So there is no capsule on the back of the lens. So that shows you, if you see this capsule, it shows you that this is the anterior surface of the lens. And this is the posterior surface of the lens, right? Here you can see a bunch of crystalline proteins. These are cells within the lens. These simple epithelial cells through life elongate and eventually become additional lens fibers, okay, through your life. With age, they constantly elongate these cells and become fibers. The problem is, what's going to happen to the lens if you keep on adding fibers? What's going to happen? The lens becomes thicker. Is it good for vision? No. Okay, so you see that eventually the older people, they reach a point where they cannot see far away, nor can they see up close, because the lens, pardon my French, are totally fucked up. Okay, so they're super thick thanks to these simple cells that are constantly adding up. It's a natural process. You cannot stop it. Okay, there is going to be a part in your life where you won't be able to see, like, not far away nor close. This is still your lens, but which surface of the lens is this? Posterior, because there is no capsule. Why lenses are so eosinophilic?
Because of crystalline.
Very good, because of crystalline. And crystalline is a
protein.
And protein stains very good with eosin, right? So it stains very good with eosin because proteins typically have a positive charge. Okay, choroid. It's a vascular layer of the eye, contains some connective tissue. It's part of the uveal tract, as I told you, right? So choroid, ciliary body, and iris together are making up a structure called uveal tract. Basically, what three of them have in common is that they're vascular. Iris has blood vessels, ciliary body has blood vessels, and choroid plexus also has blood vessels. Because they have blood vessels, what is the job? Oxygen and nutrition. What are the layers of the choroid? The top layer, the outermost, is called Holler's layer. That's where we have large diameter blood vessels. Next, Sattler, medium, and then choriocapillaries, so small blood vessels, and finishing up with the Bruch's membrane. Bruch's membrane is just your basal lamina. It's just your basement membrane in a way. So take a look. What is this? Take a look where we are, right? What is this?
Retina.
This is retina. This is the visual retina. We're in the most posterior part of the eye, right? What is this? Sclera. And between you can see that this is the choroid. So from here until here, this is the choroid. So the top layer is the Holler's layer, right? So this is the layer where we have huge blood vessels. The second layer until here, right? So how did we call it? Was the Sattlers. But not every professor agrees that this is the Sattlers. Some professors say there is no Sattlers in here, that there is simply Hollers and choriocapillaries. But some say that this is the Sattlers. The third layer is the choriocapillaries. And then I'm not sure if you can see it, there is a very thin line here, very thin line, right? So that's going to be the lamina basalis right here. This pigmented tissue is not part of the choroid. It's part of the retina. So don't confuse it. So this is already the beginning of the retina. So again, choroid, we have the Holler, we have the Sattler, we have the choriocapillaries, and this thin line right here is the lamina basalis. So 4 layers of the choroid plexus. The blackish line, this is already pigmented epithelium. So retina, 10 layers. Okay, so here they are. But I'd rather talk about them as we go. So the top layer is pigmented epithelium. It has some pigmented epithelial cells. These pigmented epithelial cells have tight junctions between each other. Okay, so that's where the blood-retinal barrier would be, right? So they have tight junction between each other. This is questionable, so not every professor agrees with it. That could phagocytosis be there? This is questionable, so in the exam you will not see this portion, but you will see that what's the purpose of the pigmented epithelium, something to do with the absorbent light. The second layer is the rods and cones, but that's not where the rods and cones are sitting. This is simply the layer where their axons will be. Dendrites, axons, okay, so like different parts of the rods and cones will be. Then we have the outer limiting membrane. It's just where additional intercellular junctions between rods and cones will be. Okay, so maybe like zonula adherents could be present here. Outer nuclear layer, that's where the nuclei of the rods and cones. Outer plexiform, synapse between them. Inner nuclear layer will contain glial cells. So Mullerian, amacrine, comma, bipolar, comma, horizontal cells. And some plexiform, but we never see them. So Mullerian, amacrine, bipolar, horizontal. So these are the glial cells in the retina, and they're sitting here. In the inner plexiform, you can see a synapse between the glial cells and ganglion cell. In the eighth, you see the ganglion cell body, the cell body of the ganglion cell itself, right? In the ninth, you see the axon of the ganglion cells that is exiting. And then sometimes you see the inner limiting membrane. It's just the basement membrane of the Mullerian glial cells specifically. So rods and cones, photoreceptors. Rods are for black and white, cones are for visual, right? There's a third type of light sensing cell. It's called the ganglion cell. It's the one which is sitting at the bottom. This is the ganglion cell, which is very important. Its axon creates the optic nerve. The axon of the ganglion cell creates the optic nerve. Now, here, I want to get your attention to the following. There is something here. Do you see? It's the lens itself, and this is where the optic disc will be. We said that the inner part of the retina, will contain ganglion cells. The ganglion cells will send their axons all the way this way to create the optic nerve. But between the lens and the optic disc, the point where we collect the axons of the ganglionic cells, there is a canal. This canal is called hyaloid canal. Hyaloid. Okay, so this canal is called hyaloid canal. In adults, hyaloid canal contains lymphatic vessels only. So it doesn't have blood vessels. It just has the lymphatic vessels. We're carrying the lymph from the lens all the way this way. In embryo, hyaloid canal contains hyaloid artery. Hyaloid artery supplies lens, of course, because that's where lens are, right? So you can see that's where canal is. What is the purpose, I repeat, of the hyaloid artery? It supplies lens. We have to make lens. We have to grow it. So in embryo, we have an artery just for the lens. It's called hyaloid artery. Okay, some additional information about the cells. We have the Muller cells, the Mullerian or Muller glial cells. They are basically glial cells. So additional information is here. You know what glial cells do, just some support, I don't know, storage, etc. Retinal ganglion cell is the most important, is the one which is at the bottom. As we have said, its axon is becoming the optic nerve itself. Who gives the information to the retinal ganglion cell? Well, the rods and cones, of course, right? So they're the photoreceptors, so they have to synapse with the ganglion cell. The problem is that rods and cones do not synapse with this cell directly. they use the glial cells. Which ones? The amacrine and the bipolar neurons. Remember bipolar neurons in our first neurohisto lesson, right? So they had 2 axons. So one axon of bipolar neuron synapses with rods and cones. Another axon of bipolar neuron synapses with this ganglion cell. So this cell is the most crucial. It has a very important job. It actually connects your rods and cones to the ganglion cell. So that's what we talked about. Amacrine cells, nothing interesting. Okay, so they're synapsing with ganglion cells, they're synapsing with another bipolar cell. The only interesting part about it that it is inhibitory. So it can inhibit the light pathway, right? So it can inhibit your retina from receiving light. Typically it happens when you're sleeping. Okay, so this cell is more important in your sleep cycle. It just suppresses the function of the retina. Okay, let's try it. So what was this?
Sclera.
This is sclera. This?
Choroid.
Choroid. The first layer is the pigmented epithelium of the retina, right? So this is pigmented epithelial cells, tight junction, etc. Second layer. This is the area of rods and cones, dendrites, axons, etc. Okay, so it's just a layer where rods and cones have some kind of cell processes. The third layer is going to be the outer limiting membrane. You can see it here. It's like a line. Do you see? Very nicely visible here. So this line, this is the outer limiting membrane. This is just zonula adherence between rods and cones. The fourth layer is called outer nuclear layer, and these nuclei are nuclei of rods and cones. These, okay, they're the nuclei of rods and cones. The fifth layer is going to be the outer plexiform layer. In the outer plexiform layer, you see that these rods and cones are going to be synapsing with the glial cells. A very good example of the glial cell that synapses with rods and cones in the outer plexiform layer is a bipolar neuron. Okay, bipolar neuron will be synapsing with rods and cones here. The fifth layer is going to be the inner, not the fifth, the sixth. The sixth layer is the inner nuclear layer. In the inner nuclear layer, that's where the nuclei of the glial cells are, right? So the amacrine, the bipolar, the Mullerian glial cells, they're sitting right here. The next one is going to be the inner plexiform layer. The inner plexiform layer is where the glial cells are synapsing with the ganglion cell, right? So in here, in the seventh inner plexiform layer, we have a synapse between the glial, like bipolar, and the ganglionic cell. The eighth layer is the ganglionic cell layer. So these nuclei, they belong to the ganglionic cells. The ninth layer is the nerve fiber. This layer right here, it contains axons of the ganglionic cells. So these are axons of the ganglionic cells. They're starting to create the optic nerve. The 10th layer is the inner limiting membrane, but you cannot see it here. There's a reason why I only put 9, right? So the 10th layer, the inner limiting membrane, you cannot see it here. It was lost. You can see it here. There's a bit of a line here, and this line is the inner limiting membrane. This. Okay, so this is the inner limiting membrane. So let's do it again. Sclera, choroid, pigmented epithelium, rods and cones, nicely visible outer limiting membrane, outer nuclei, outer nuclear layer, outer plexiform layer. inner nuclear layer, inner plexiform, ganglionic, nerve fiber. Tell me what happens in this layer. What is this? What are these?
Nuclei of rods and
Yes,
cones?
these are nuclei of rods and cones. Whose nuclei are these?
Glial cells.
Glial cells, such as bipolar, amacrine, Mullerian. Whose nuclei are these? Ganglionic cells. Here you can see nuclei as well, like this one, this one. Whose nuclei are these? These nuclei belong to
Schwann cells.
Schwann cells are in the peripheral nervous system. The
Oligodendrocyte.
I could see why you would think so, but no. These cells are the Mullerian glial cells because their axons will create the inner limiting membrane. Inner limiting membrane has something to do with the Mullerian glial cells. So these dots, they are nuclei of the Mullerian glial cells. Okay, the same idea here. Okay, so sclera, choroid, bunch of layers of retina. Just don't forget here, you can actually see the inner limiting membrane. Okay, finally, let's do the whole slide. I point, you tell me. Okay, what is this?
Cornea.
Cornea,
Anterior chamber.
anterior chamber,
Iris.
iris,
Posterior chamber.
posterior chamber,
Ciliary body.
sorry, all the way until here. Okay, so this is the ciliary body right here. This is
Lens.
lens. Okay, what should be here?
Vitreous humor.
Vitreous humor. Okay, what is here?
Sclera.
Sclera, this little layer,
Choroid.
choroid, and
Retina.
retina. This
The optic canal.
disc, and from here the optic nerve will begin. Okay, cornea, how many layers?
5.
So epithelium, anterior limiting membrane Bowman's, stroma, posterior limiting membranes deciment, endothelium. How do we call this angle?
Iridicocorneal.
Iridicocorneal angle. It's between the cornea and the iris itself. These.
Ciliary body.
Thank you so much, Pooja. Now somebody else.
Ciliary muscle.
Ciliary muscle. Okay, what are these holes? This one and this one.
Space of Fontana.
Spaces of Fontana are here. They're in the area of the iridicocorneal angle. Okay, so that's spaces of Fontana. What are these? Ocular veins. These are the ocular veins. Canal of Schlemm will be somewhere here. Okay, so yeah, so canal of Schlemm's are here. Maybe this one also would belong to canal of Schlemm, but closer to the limbus area, right? So you're gonna see that's where the ocular veins would be. What is this?
Bulbar conjunctiva.
Bulbar conjunctiva. So fornix, sclera, choroid, retina. You get the idea. Let's do the aqueous humor pathway quickly. So one is
Posterior chamber.
posterior chamber. 1A is
Iris.
Pupil. Okay, we cannot see it here, but somewhere through the pupil, right? So somewhere here, it's the second part. Okay, 2 is
Anterior chamber.
Anterior chamber. 2A is going to be
Iridicocorneal angle.
3 are
Space of Fontana.
spaces of Fontana. 4
epithelial anastomosis,
Endothelial anastomosis, 5 V's.
canal of
Schlemm, 6 are somewhere there, we cannot see it, maybe this part belongs to it. These are the ocular veins, and the last one is going to be here. 7 is here. Central retinal vein, yeah. Okay, it's going to be where the optic disc is. Is it clear?
Yes.
Okay, sclera is going to be somewhere here. We have a bit of a choroid and retina. This area right here is the optic disc. Optic disc will become the optic nerve. Sclera is continuous with the fibrous sheath of optic nerve. So somewhere here, the outer part, that's the fibrous sheath of the optic nerve, and they're together. They're continuous with one another. Okay, the sclera and the optic nerve sheath. You can see in the optic disc that there are 2 vessels here, very nicely visible. Vessel number 1, vessel number 2. We magnify. This one is going to be the central retinal vein. This one is the central retinal artery. How do I know that this is the artery? It's quite thick, right? You can see that the wall layer is quite thick. It has the muscular component. The vein looks a bit collapsed, so it doesn't have the muscular component. Okay, the same idea, we did the slide several times, right? So the sclera, the choroid, just tell me, how do we call this layer of choroid? Hollers, second. Sattler. Choriocapillaries, and then there should be a line, but we cannot see with this magnification. The line should be the lamina basalis, right? That's the 4 layers of the choroid. Top layer is sclera, bottom layer is retina. Okay, let's finish up some things related to eye. So here we have the lacrimal gland. Lacrimal gland, its function is to make tears, right? To secrete tears. It's a serous gland, but its cytoplasm is quite eosinophilic due to different ratios of RNA and proteins. It's a serous gland, so it has a bunch of ducts. The ducts are going to be located in the connective tissue, but they don't really have a stratified appearance. Typically, whenever you talk about the gland, try to remember the first semester, we always saw stratified cuboidal or columnar epithelium, but that's not the case here. Here you would see either simple or pseudo-stratified epithelium. Type of secretion, merocrine. Tears contain a bunch of antibacterial materials, right? So to make sure that you clear the surface of the eye. And yeah, okay, so that's your lacrimal gland. Honestly, nothing interesting. It looks like a normal serous gland. These are the serous acini. This is the connective tissue. This is a duct, right? So that's another duct. The difference between this duct and this duct is that one of them is intralobular, one is interlobular, as you remember. The one which is surrounded by connective tissue is interlobular. From last semester, you should remember that this is called septa. And septa, the pink tissue, makes your lacrimal gland lobulated, right? You can see lobules very clearly. Boring stuff. So serous acini, intralobular ducts, interlobular ducts, intralobular duct, intralobular, right? The only difference is whether you have connective tissue septa surrounding it or not. That's it. Now, this is important. Okay, so let's talk about the eyelid itself. Let's do layers. So how do we call this tissue?
Cornea.
Cornea. Behind it is anterior chamber, that's iris. Behind it is posterior chamber, lens, suspensory ligaments, ciliary process, ciliary body, sclera. You get the idea. On the surface of the sclera, we can see that there is conjunctiva, right? This is the bulbar conjunctiva. How do we call the area where we reflect the conjunctiva? This is called the fornix. Let's do the layers of the eyelid from the most outer part. The most outer part of the iris is, of course, skin, right? It makes sense. So skin is this area right here. This is the epidermis. Right under it is going to be the dermis. Right under the dermis of the skin of the eyelid, we have a muscle. This muscle is known as the orbicularis oculus muscle, right? It's a circular muscle that goes like that. The innervation is the facial nerve of the orbicularis oculus muscle. Which kind of muscle it is? It is a skeletal. Why? Because I can do this, right? So I'm in full control of my eyelids. I can blink on if I want to. Beneath the orbicularis oculus muscle, we're going to have at first loose connective tissue. So this area right here, it will be loose connective tissue. And then suddenly this loose connective tissue becomes dense. Within dense connective tissue has a name. It's called tarsal part. So this area right here, this is known as the tarsal part. Within the tarsal part, you have a very special sebaceous gland. It's huge. So inside of the tarsal part of the eyelid, there's a huge sebaceous gland, which is called meibomian gland. It is a sebaceous gland, so its type of secretion is holocrine. Under the meibomian gland or under the tarsal part, you're going to have palpebral conjunctiva, right? So the conjunctiva, which is reflecting to become the bulbar. There's some additional sebaceous glands present. They're called Zeiss. Zeiss glands are associated with your eyelashes. Okay, so they're right there surrounding your eyelashes. Between the meibomian sebaceous gland and gland of Zeiss, between them, there are apocrine glands of Moll. So somewhere here, you're going to see the apocrine glands of Moll. What is the purpose of Zeiss, meibomian, and apocrine glands of Moll? Lubrication. Okay, so they're secreting this fluidy substance, which is going to be coating your cornea, coating your bit of sclera here to make sure that your eye does not dry out. The one job it has is definitely the lubrication, to keep the surface moist. Okay, we have two muscles. Okay, we're gonna have two muscles in this region. Additional muscles. In addition to the orbicularis oculis, you're going to see that we have the levator palpebral muscles and superior tarsal muscles. They're going to be somewhere here on our slide. Okay, so if I go here, so there are going to be two additional muscles, the levator palpebral and the superior tarsal muscle. Out of the two, levator palpebral muscle is the skeletal muscle, superior tarsal is the smooth muscle. Levator palpebral muscle has a tendon which attaches to the tarsal part. Remember we said tarsal part is the dense connective tissue right here. This dense connective tissue contains meibomian gland. So the tendon of the levator palpebral will be attaching to the tarsal part right here. Let's do the layers. So first of all, this is your eyelid. How do you know that this is the superficial and this is the inner? How do you know?
Skin. If there's a skin. Yeah.
Very good. Exactly. Skin. This surface right here, this is your epidermis. Beneath this area, this is dermis. So this is epidermis, this is dermis together, this is your skin. What is this? Palpebral conjunctiva. You follow palpebral conjunctiva, you follow, it stops. How do you call this?
Fornix.
That's where it should reflect to become the
Bulbar conjunctiva.
bulbar conjunctiva, yes. So here, this should have been the bulbar conjunctiva right here, but that's the fornix. Okay, so beneath the dermis of the skin, there is a huge skeletal muscle here. How do we call the skeletal muscle?
Orbicularis.
Orbicularis oculus, right? So oculi or oculus is huge skeletal muscle. Beneath it, you can see loose connective tissue. Right, this is loose connective tissue, and then loose becomes dense. You see, lots and lots of collagen fibers are here. This is loose connective tissue. This is dense connective tissue. So this area right here is known as the tarsal part. Okay, so this area is the tarsal part. How do we call the gland in the tarsal part? Meibomian. Okay, so this is known as the meibomian sebaceous gland. So this is the meibomian sebaceous gland. It's the biggest sebaceous gland there is. These are apocrine glands of Moll. These ones. These are hair follicles for the eyelashes. And I know you cannot see it, but here we have sebaceous glands right next to them. These sebaceous glands right next to the eyelashes are called glands of Zeiss. So meibomian, Zeiss, between them, the apocrine glands of Moll. At the top here, we have two muscles. There's one muscle here, and there's one muscle here. Okay, so one muscle here and one muscle here. The bottom one is the superior tarsal. The top one is the levator palpebral. How do you know? How do you know that the bottom is the superior tarsal, the top is the levator palpebral? What do we need to check for?
Bundles?
Bundles, we have to remember how does the skeletal muscle looks like, right? So we're going to see that this top is going to be the levator palpebral, the bottom is going to be the superior tarsal, it's the smooth muscle. But we'll get back to it in a sec. Okay, again, what is this? Now we're going from the opposite direction. Palpebral conjunctiva. This, like starting from here, finishing here. What is that? Tarsal part. Tarsal part is basically dense connective tissue. You can see it here. There's a bunch of collagen fibers here. Within the tarsal part, we can see this gland.
Meibomian.
Meibomian. Which kind of gland is it?
Sebaceous.
Sebaceous, very good. It is a sebaceous gland. So its type of secretion is holocrine. Do sebaceous glands have ducts?
I think it was an exception for
Exactly. Meibomian gland is the only gland that has a duct. It's the only sebaceous gland that has a duct. Because the type of secretion is holocrine, you don't need a duct. You simply kill yourself and all your secretions are everywhere. That's it, they're everywhere now. But meibomian is the biggest sebaceous gland, so that's why it definitely needs a duct. We'll talk about this duct in a sec. How do you call this area?
Loose connective tissue.
Very good. This is the loose connective tissue. What is above loose connective tissue?
Dermis.
Between loose connective tissue and dermis, there is orbicularis oculus. Okay, so this is going to be the loose connective tissue. This is the dermis. Between loose connective tissue and dermis, there is a muscle, the orbicularis oculus muscle. So this duct is very special because it can get inflamed, and the inflammation of the duct of the meibomian gland and the rest of the meibomian gland is known as the stye. Okay, so this is very painful, okay, very itchy. You can see that there is pus. Okay, so this girl is lucky because this is on the surface. It can be on the inner part, so it's bugging the eye, you can't blink, and etc. Okay, so STI is the inflammation of the duct of the meibomian gland. Why is it inflamed? Because basically, do you see this is the duct? It's free to the outside. Like you didn't wash your hands, you itched your, I don't know, eyelid, you introduce bacteria this way, they are proliferating in the duct of the meibomian gland, inflammation, that's it. So STI. Let's do it again. So this is epidermis, dermis, orbicularis oculis, right, which is the skeletal muscle, clearly visible here. We have a bunch of bundles. Loose connective tissue, tarsal part, which is the dense connective tissue, meibomian gland, and conjunctiva. To be more specific, the palpebral conjunctiva is right here. So at the top, I told you we have two muscles. This one is the superior tarsal muscle. This one is the levator palpebral. How do I know? I need to check the bundles. Even with this magnification, I can see the bundles of the skeletal muscles. I can clearly see them here. While this muscle is like a spindle shape, it's more elongated, more stretched out. To prove it, we magnify, right? So we see bundles of the skeletal muscle, levator palpebral. We see elongated smooth muscle cells, superior tarsal muscle. Yes, two muscles here. Which one attaches to the tarsal part out of the two?
Superior tarsal.
Levator palpebral. It has a tendon, and this tendon will be attaching to the tarsal part. Take a look where we are, right? So this is the conjunctiva, the palpebral conjunctiva. There's a lymphatic nodule. This lymphatic nodule is proof that this eyelid is inflamed. Okay, it's not normal to have this lymphatic nodule here. It's not normal. So it's a proof that there is some kind of infection going on. There's some kind of inflammation in this eyelid. We have accessory lacrimal glands, these ones. They're called glands of Krause. So we have the major lacrimal gland, the one that we did, the huge serous gland, right? And we have some accessory in the eyelids. They're called glands of Krause. So again, what are these?
Sebaceous gland.
Sebaceous meibomian glands, right? So these are the hair follicles. They are the eyelashes, right? So the eyelashes will start from here. How do we call the sebaceous glands associated with eyelashes? Zeiss, yeah. So these are going to be the glands of Zeiss, these little ones. As you can see, nothing special. They're still sebaceous glands. Just the only thing that is special is that they're next to the eyelashes. And these between the meibomian and the Zeiss, you're going to see the apocrine glands of Moll. How do you know that they're apocrine? The lumen is huge. Maybe you remember the apocrine sweat glands. They are sweat. It's like my favorite saying ever. Russians don't cry. We sweat from our eyes. It's actually true. Okay, so it's actually true. So these glands produce sweat. And this sweat is very rich in pheromones. Remember, like in axilla, we also have apocrine sweat glands. They're responsible for pheromones, for attraction. Of course, the purpose of the glands of Moll is not to attract the opposite sex, like for axilla or anal or pubic glands, but more for lubrication of the eyelid. But they're still apocrine sweat glands. So hypothetically, they're still able to produce, secrete pheromone. Yeah, as you know, pheromone is the opposite sex attractant. Okay, so eyelashes, glands of Zeiss. That's it. Okay, so we're gonna finish here. We're gonna finish with the eye. And I think we have decided that I'm going